Welcome. I'm very pleased to introduce Shirley Liu. She's a professor in the Department of Data Sciences at Dana-Farber Cancer Institute and a professor at Harvard T.H. Chan School of Public Health. She's also the director of the Center of Functional Cancer Epigenetics at the Dana-Farber Cancer Institute. Her computational biology lab delivered tools in transcription factor binding motif finding, ChIP-seq and ChIP-chip chromatin accessibility profiles, CRISPR screens, and tumor immune interaction. She's a fellow of the International Society of Computational Biology, a recipient of the Sloan Research Fellowship, an ISMB and an ISCB 2020 Innovator Award, and a recipient of the 2020 Benjamin Franklin Award for Open Science and Open Access in Life Science. And I'm delighted to introduce Shirley today.
Yeah, thank you, Aidan, for that kind introduction. It's very exciting to present our work. This is a new direction. We're trying to model protein degradations in cancer. So here's my disclosure slide. I won't actually discuss anything right now with it, any of this company-related things. Okay, so the human has ubiquitin proteasome system. It basically degrades most of the proteins in the human cells. So basically, if you have a cell, and usually the sequence that's being recognized by the UPS system is this background sequence, which is usually in the end, either the N terminus or the C terminus of a protein. And a UPS complex, this E3 complex, would recognize the background sequence and then bring the E2 to add ubiquitin onto the end of the protein. And this ubiquitinated protein will be targeted for going to the new proteasome. And this really shapes like a trash can. And the ubiquitin will just go in and thread the protein through. And when it's outside, the protein will be degraded. And the ubiquitin can then be reused to degrade the next protein. This is an equilibrium because there's another enzyme, the deubiquitinating enzyme, which basically removes the ubiquitin to make the protein more stable. So the E3 and the deubiquitin enzyme, they kind of work against each other. Over the years, there have been many drugs that are developed. Some targets the proteasome, which makes the protein more stable. Others targets the deubiquitinating enzyme, making the protein more towards degradation. But the most exciting work in recent years is something called PROTAC, or proteolysis targeting chimera. Basically, on the left side, what you see is this big E3 complex. And so this, the ubiquitin complex, this is E3, there's a scaffold, and here's the E2, and they recognize some substrate. So the idea is basically, initially, this E3 may not really need to recognize the kinase itself. Instead, we can use a small molecule to string the two together. One end of the small molecule can bind to the target substrate very strongly. The other end of the small molecule can really target this E3. So with this small molecule, you can bring the E3 UPS system to a protein you want to degrade. And once they are close, the E2 will add the ubiquitin onto the kinase. And very often in 15 minutes time, the cell will be depleted of the protein that we want to degrade. This is really exciting. In 2019, there's a Nature News and Feature article showing that PROTAC can drug the undruggable. Basically, the PROTAC is a small molecule. You can see one end targets the protein, the other end targets the ubiquitin ligase, and once they are close to each other, the ubiquitin will be added to the target protein for degradation. You can see here the proteasome is really quite like a trash can. And the nice thing is really previously, in order to use small molecule to target a protein, the small molecule really have to uniquely bind to the target protein at the direct activation or enzymatic pocket. But with a PROTAC, we can just degrade the whole protein. So as long as the small molecule can uniquely bind to the target protein, they don't really have to target a specific enzymatic pocket. And so the really exciting possibility is that we can potentially degrade transcription factors because many transcription factors are really misregulated in cancer. And so because they don't really have an enzymatic pocket, it's very, very difficult to make a small molecule to target them. But with PROTAC, now we can drug the undruggable. And since I have been working on transcription factor regulation in cancer for many, many years, this is an area we really want to get in. And so over the years, the work that we thought was interesting is, or whenever we target a new direction, we want to make sure that it's a direction that we can leverage our existing expertise, then we can move to the new direction. And in addition, Colin Tokay is a new postdoc was a new postdoc in the lab at the time. And during his PhD years at Johns Hopkins with Rachel Kachin's lab, she worked with TCGA pan-cancer analysis for all the genes that are frequently mutated. And in this figure, the very bottom are the most commonly mutated protein or gene pathways, and then the top ones are the gradually less and less frequent pathways. And you can see here, the most frequent ones are kinase pathways. And so there are, recent years, we see plenty of kinase inhibitors developed. There are also things like genome integrity or cell cycle. These are usually targeted by chemotherapy. And then you see the chromatin, histone modification, chromatin histone modifier. So there are a lot of epigenetic inhibitors that are developed. And we also see immune signaling. These are immunotherapies. But you can see transcription factors and protein ubiquitination. It's very frequently mutated, but currently there hasn't been good drug that can really target these two pathways. So we are trying to use PROTAC to really look at the two together. This is our general computational approach. So this is Colin's main work in here. We look at all the TCGA cancer types or all the tumors. There are two things we look at. One is on the mutations on the protein that's responsible for degrading other proteins. So these includes the E2, E3 ubiquitin ligase, and also the D-ubiquitin enzymes, whether they have mutations and whether that has some downstream consequences. This is looking at mutation of UPS in trans, and then we can also look at mutations on the substrate of those E3 ligases, which are usually at the N-terminal or C-terminal ends of the proteins, and these sequences changes the recognition site from the UPS proteins, which will make the protein more or less stable. And in this end, we also use a deep diagram inference model to look at how would these diagram mutations influence protein stability. In addition, we try to see whether mutations on the UPS protein influence the downstream substrate, which are transcription factors. Then it has even stronger effect by perturbing the transcription factors downstream targets. And so that's the general computational approach. The first approach is to look at direct UPS protein mutations. And so for this, Colin has a PNAS paper that he developed during his PhD in collaboration with Bert Vogelstein's lab at Johns Hopkins. And they call it 2020+. And basically, based on the gene expression and mutation patterns, they can assign each gene as a probability of being either an oncogene or tumor suppressor gene or a passenger mutation. And so based on this, we can also look at we are restricted to, in this case, just looking at the ubiquitin and the D-ubiquitin enzymes, and to look for those that are statistically significant in recurrent mutations in TCGA. And the overall analysis of here, we can see we identify many, many recurrent mutations that are on the UPS pathway, really accounting for 16% of all the cancer driver genes, which is quite amazing that such a significant portion of cancer recurrent mutations are related to the UPS pathway. And so compared to previous analysis from other studies, as well as a TCGA pan-cancer analysis, the 2020 plus can identify 33 previously not reported driver mutations in the UPS pathway. When we look at the presence of these genes, majority, 3/4 of these genes are related to the E3, which is responsible for recognizing background sequence on the substrate. And 17% are related to de-ubiquitination, which removes the ubiquitin. And then we have the E1, E2, which is, E2 is related to adding the ubiquitin onto the protein for degradation. Next, we look at what are the known diagrams that are documented in the database. So we look at a known diagram database, which only contains diagrams of 200 proteins. And so supposedly this is the known real diagram. We will just randomly simulate a fake diagram, which is just another random sequence of the same length. And then we use just 200 genes to train a small kind of a machine learning method. And so previously, Rachel Kachin's lab has a toolbox called SNV box. It basically annotates proteins. For each position on the protein, it annotates an evolutionary conservation. It also annotates whether this location is in a more flexible region of the protein, for example, not in alpha helix or beta strand, but in more of a loop region. Also annotating whether the protein is in a residue that's kind of more solvent exposure facing the outside rather than packaged inside of the protein. Also looking at hydrophobicity and also charged amino acids. And so Colin built this random forest to see, based on the known real diagrams, can we see what features are important for diagrams? And you can see, based on the cross-validation, even just looking at very simple features, we have some predictive power, although the prediction accuracy is only at 0.181, which is not perfect. But you can see the most, in terms of feature importance, the number one importance is evolutionary conservation. And then it's flexibility, and then amino acid solvent exposure. Basically, diagrams tend to appear in evolutionary conserved region that are in loop regions that are also facing the outside of the protein that are kind of solvent exposed. And so, so we have a big list of this, and one example of a cancer driver gene is cyclin D1. You probably know that cyclin D1 is very important for regulating cell cycle. And if we look at the cyclin D1 mutation patterns in here, you can see most of the mutations happen towards the end. Normally, if you have loss of function mutations on the protein, they should be kind of scattered around on the protein. But when you see something really quite enriched in the end, this usually suggests that this is related to the diagram. Indeed, if we look at the known diagram sequence here, a lot of the mutations potentially change the protein status. And in TCGA, the good thing is that TCGA has the RPPA protein microarray data, at least to measure cyclin D1 in many tumors. And so if we look at those cancers that has wild type cyclin D1 expression, protein level, versus those that have either the non-silent mutation or this missense mutation, which will actually influence the phosphodiagram sequence, you can see that when the mutation happens to perturb the phosphodiagram sequence, actually on the RPPA, this protein is indeed much more stable, and it can accelerate the cancer cell for proliferation because it's a cell cycle regulator. This is another example of a diagram loss. This is related to Gleevec, this drug that targets CML cancer, which has a typical BCR-ABL fusion, BCR-ABL fusion. So you basically, the beginning of the BCR gene is fused to the second half or the later half of the ABL gene. And interestingly, in this case, most of the fusions previously we believed were basically the later half really drive cancer growth, and the first half drive a tissue-specific high expression. And so with this fusion gene, you can have higher expression of the second gene. But in this case, interestingly, there is a diagram at the beginning of this protein, and this fusion actually happens right after that diagram. And AsCOP is the E3 ligase that's responsible for recognizing this diagram and degrading ABL protein. And when we look at the CML patient with this mutation, not only we see overall higher ABL gene expression compared to a regular ABL expression due to the BCR giving the correct or the activated promoter, but also the protein is more stable because of the loss of this diagram. So even though we can have some predictive power of diagrams just based on evolution, flexibility, and solvent exposure, because the database of all the diagrams has only 200 diagrams, which is a very small data set. So our overall predictive power isn't so great. And so in order to improve this, we looked at a 2018 Cell paper published by Steve Allergy's group. So in this study, they developed an assay called Global Protein Stability proteomics assay. The idea is they first use oligosynthesis to really synthesize the RNA or the DNA, like expression vector, for all the human proteins in the last 23 amino acid sequence. And so basically, they have a vector of the end of every protein, so the protein coding amino acid 23 sequence peptides for every protein sequence, and they clone those into an expression vector. And so within here, you see this very strong promoter, and it first express a red fluorescent protein, and then there is another green fluorescent protein, then followed by the peptide. This peptide is the N23 amino acid of every gene. So in different clones, it will be a different peptide sequence. And these are actually transcribed as two separate proteins. The red protein serves as a control, whereas the green protein will be expressed, but it can be degraded if the peptide sequence contain a strong degrader or diagram or a weak diagram. So they could either be a very strong GFP or a weak GFP. And so when they use this pool of lentivirus to package this lentivirus into a pool and then transfect the cells, then each different cell will take up a different lentivirus to express. They all express the red protein. They also all express the green protein, but depending on the peptide sequence at the end, the green protein can be degraded at different rate. And so based on the fluorescent sort, they can decide whether this protein, the peptide sequence, is a stable diagram or unstable diagram. And so towards the top, basically the GFP and the red fluorescent are all very high, so these are more stable. Whereas in the bottom here, the red protein signal are very strong, but the green, the GFP signal is very weak. These indicate an unstable protein. And so the nice thing here, we have now 20,000 proteins C-terminal ends to do the modeling. And so for that, we then build a deep learning model. We call it deep diagram. And so we didn't do really an overkill. And we only have 2 hidden layers in here to predict whether this is a degradation high or a degradation low class. And the input is basically the peptide sequence in the GPS vector, the 23 amino acid sequence of every human protein. And output is to predict whether this is high or low protein degradation from the GPS. And using this deep learning model, we can actually very well predict the GPS assay. And also, when we really look at a specific diagram, we try to look at 2 things. The very naive model only look at amino acid sequence, because even just looking at the overall amino acid makeup, you could make some guesses. There are some existing rules for, for certain amino acid to be more likely to be degraded, but we are more interested in a longer sequence and also a sequence-specific degradation. So when we use the position-specific degradation subtracted by just a bag of words of amino acid, we get the real diagram potential. And using this approach, you can see here with cross-validation, you can see that deep diagram really have a much better prediction of the correct degradation rates of different C-terminal, protein C-terminal peptides. Compared to, I guess, random forest still works reasonably well compared to like logistic regression or just, like just amino acid, we actually get worse predictive power, although it still can predict a little bit. And so from this approach, we identify some diagrams that were previously not known. So the original database has only 200 known diagram cases. So in this case, we actually identified a lot more diagram sequences, including some that are, these two positions flanked by, by another amino acid in the middle. So there's some gaps in the middle, but still we can predict those type of diagrams. And so we reached out to Steve Allergy's lab, and they told us that in addition to the first cell paper they published, they had a second cell paper which had, so the initial paper was on the C-terminal end. They had another paper on the N-terminal end. We also used the diagram to make predictions. And for all the novel diagrams that we identified, we contacted them for potential experimental validation. And so they already have the GPS assay set up. So they actually try to see if the diagrams are mutated from the original wild type to the sequence that we predicted, whether that will make the proteins more stable. So the black are the original sequence with the diagram, but when we change the amino acid sequence, which basically will disable that diagram sequence, and you can see in each of these validation cases, the protein did become more stable because the diagram sequence is lost. And so with the novel diagrams that we can predict using deep diagram, we can then apply this to all the somatic mutations of TCGA tumors to see which tumors will have a diagram sequence that's mutated on the protein, which will make the protein more or less stable. We also use a background model, which we just randomly mutate proteins and look at how often those change the diagram potential. And you can see the background distribution is kind of close to zero, whereas if we observe a particular true occurrent mutation that's really changing the diagram mutation, we can assign a statistical significance to this observation. And so based on this, we look at also, again, pan-TCGA tumors, and the most significant diagram mutation, these two are the strongest. One is a mutation at the C-terminal end of GATA3 protein in both breast cancer and also pan cancer. The other is this gene PPM1D. So let's first look at GATA3. If you look at GATA3 mutations, this is also in breast cancer. You can see again, most of the mutations actually happens at the end. And also interestingly, GATA3 also have RPPA data in TCGA. So we're lucky, even though RPPA only profile a few hundred proteins, they do have the GATA3 protein on it. And so you can see a lot of these mutations are actually frame shift mutations, right? Happening here. Usually when you have a frame shift mutation, you would believe the remaining protein will be a total garbage, and this protein might get degraded from nonsense mediated decay. But in this case, because the most of the functional domain on the GATA3 is still there, and the frame shift mutation pretty much only lost the diagram. And in this case, we can see the GATA3 mutations with a frame shift mutation actually made the protein much more stable. We also specifically mutated the actual diagram sequence that we predicted. For example, this is the diagram motif on GATA3, and when we mutate A to an M, and then also we try to mutate this G into a E, and in each case we want to see whether this will change protein stability. So in this case, it's not really a frame shift mutation. We just made a point mutation directly on the diagram sequence. And you can see here, this is the wild type, and this is the A to M mutation. This is the G to E mutation. And other ones, we can also use a flag tag. This is a control. When we add a flag tag, basically, if the protein previously ended in here, now you add another sequence, then the background is no longer being recognized. You can see in the control, definitely the protein is more stable, but also even in the point mutations, we can see the proteins being more stable, suggesting that we are predicting the correct diagrams on the GATA3 protein. Next, we did the ChIP-seq because the GATA3 is a transcription factor. We wonder when you have a more stable transcription factor protein, what's the downstream consequence? And so then we did ChIP-seq of GATA3 using the wild type protein and also the A to M and the G2E, more stable protein. And in each case, we see compared to the wild type protein that GATA3 is binding much, much stronger to the chromatin. And when we use this overlap of about 4,700 peaks, we look at where are they near the upregulated genes in terms of gene expression, and we see basically the pathways that are enriched near these genes that are related to the stronger GATA3 binding as estrogen signaling pathway and also proteolycans in cancer, basically cancer-related pathways. This really suggests that the frame shift mutation that we observe in GATA3 influence the GATA3 diagram and make this GATA3 more stable, which in turn activate a lot of estrogen receptor and cancer-related genes in breast cancer. And so this kind of help us interpret this GATA3 mutation, which is the second most frequent mutation in breast cancer. Another example we saw is this PPM1D mutation. This is a negative regulator of P53. And if you look at the mutation on this protein, again, we see the mutations to be enriched in the C terminal end. And again, we use the point mutations here to change the position 605 from a C to a W, or 604 from a V to a Q. And then we also try this L450 mutation in here. And so you can see in each case, this, by the way, this is a truncating mutation, which basically deleted everything afterwards. And you can see the protein is shorter now, but it's definitely more stable compared to the wild type. And interestingly, when we have these mutations, you can see even though in the cell, the total P53 levels are just across different conditions are the same, we do see that the phospho P53 on the serum 15 position become less and less stable. And by the way, for P53, only the serum 15 phosphorylated site are the activated P53. Because basically, a lot of times you have a protein, when it's phosphorylated is when it's most active. But actually, E3 ligase not only recognize diagram, having the phosphorylation also stimulate E3. So when a protein is most active, it's also most vulnerable for protein degradation. And we definitely see that in this case, suggesting that PPM1D mutation increase the protein stability. And then that basically will make P53, the functional P53, less stable in the cancers, which will make those tumors having a lot more DNA damage in the cancer cells. This is our approach looking at mutations on diagram, and a lot of the downstream consequences you can see. We try to understand it in terms of protein stability and from RPPA data. Unfortunately, as we mentioned, in TCGA RPPA, there are only about 200 proteins involved, a few hundred proteins available. Can be more like 500 proteins. And so most of the proteins actually don't have RPPA data. And so we're wondering, are there cases like this when you have a transcription factor that is a substrate of either E3 or deubiquitinase enzyme? And so based on TCGA, maybe first of all, this transcription factor needs to be expressed. There is an RNA expression level, but then it's the protein level that's really important for binding to DNA and regulating the downstream gene expression. Just based on TCGA gene expression data, we will not really be able to tell that the protein is more stable or less stable at the protein level. However, because the transcription factor regulates downstream gene expression. If we have the ChIP-seq data for the transcription factor, and we can use the downstream target gene expression to infer the transcription factor activity and protein level. And so we wonder, for example, if you have a mutation versus wild type on the ubiquitin ligase or the deubiquitinase, where the transcription factor expression is fairly stable, but because they are being degraded faster or slower, we actually see the downstream genes being different. And using this approach, we're hoping to predict which ubiquitin ligase or deubiquitinase target which transcription factors for degradation. So this will help us infer the substrate. So previously, my lab developed a computational algorithm called Rabbit. We basically look at all of the TCGA data. This has already been normalized for DNA copy number change or DNA methylation effect. So we know that previously we modeled this as everything is based on just gene expression, and based on the target gene expression, we could figure out what transcription factor might be regulating what target genes using ChIP-seq. But in this case, we are looking at all of the TCGA data to see if you see mutations in the E3 ligase or deubiquitinase, do you see changes in the downstream gene expression? And so using this approach, we were able to predict the transcription factor that are being targeted by the different UPS genes on the protein degradation pathway. And so you can see here, these are top 100 mutations. For example, this FBOX7 is the strongest, and you see many dots, each dot is being significant in a different cancer type. And so this is kind of the strongest, and then this is second, and so on. So if you look at the top 100 genes, if we were to control FDR, we believe that these top ones are all real drivers of downstream transcription factor effect. And interestingly, out of the ones we predicted, many are known. For example, FBOX7 is known to target MYC protein stability. This one is SPOP regulating androgen receptor stability. And the KEAP1, COL3 E3 ligase regulates NRF2 protein stability. Cyclin D1, CYLD regulates NF-kappa B signaling. And also the VHL regulates HIF1 alpha stability. And so there are some known ones like this. For example, KEAP1 is the E3, COL3 is the UPS, the E3 ligase, the scaffold protein. And in TCGA, when we see KEAP1 to be mutated, definitely we see the differential expression of the NFE2L2 downstream target. Even though from the expression, NFE2L2 does not have expression, RNA expression changes by itself. Also, there's no proteomics data, but we can still infer this from the downstream substrate, the downstream target of NFE2L2. Interestingly, when we see the, also there are mutations on the COL3, which is this scaffold protein. The scaffold is connecting the E2, E1, E1, E2, and E3 together. And when we see the COL3 mutation, not only we see the downstream target gene of NFE2L2 being perturbed, we also see MYC and the BRD4 downstream target being perturbed as well, suggesting that there is another E3 which can use COL3 as the scaffold to target BRD4 and the MYC for degradation. And so we experimentally validated this by using co-IP, and you can see basically COL3, MYC, and KEAP1, they kind of they are in the same protein complex, suggesting that COL3 can interact. So in this case, KEAP1 is with NFE2L2. We don't know what this other protein is. We have to kind of use the co-IP, but maybe with a mass spec to figure out what this question mark E3 is. But at least we can see that COL3 indeed is involved in the degradation of MYC protein. So the first part of the summary is that the current known information about UPS regulation in disease is limited due to the lack of systematic models. And we use unbiased genomic approach, which is this global protein stability assay, and the machine learning approach to review mutated backgrounds that are happening in cancer. And from GATA3, I hope we can convince you that truncating mutations can be gain of function because of background loss, and also the BCR-ABL fusion. Also, you can see a truncation at the beginning of the protein can also create a gain of function effect because of background loss. And together, for the UPS mutation or the background loss, we see over 19% of cancer drivers as related to the UPS system. And mutations that increase protein stability are potential targets for drugs that can induce targeted protein degradation, which we are now working with the Dana-Farber Protein Degradation Center to develop better computational tools to better develop backgrounds or the PROTACs. In the second part of the talk, I want to show you a collaboration with an experimental team. And so in the previous computational work, we have Xiaoqing Wang, who is a postdoc in our lab jointly with Miles Brown's lab to validate a lot of our computational predictions. I mean, this collaboration actually came organically because at the same time, Colin was also helping Xiaoqing with a related protein degradation project. And so for this, we want to introduce a in vivo CRISPR screen technique. Basically, we develop a CRISPR library. We infect the different cancer cells. 41 is breast cancer, EMT6 is another breast cancer case, MC38 is a colon cancer. And so in each case, different mouse cancer cells will carry a different CRISPR to knock out a different gene. And then we implant this into the mouse tumor. Actually, in this case, we also have the in vitro just cell growing in Petri dish compared to the cancer cell grown in nude mice, immune competent mice. And this is immune competent mice, but we also, before the cancer cell injection, we gave the mouse a immune vaccine so that they can induce a stronger immune response. And in the last case, not only we have the immune vaccine, we also have a PD-1 treatment to really kind of kill the tumors. And so in each case, after the tumor are grown for a couple of weeks, we take out the tumor, isolate the CRISPR in each cell, and do high throughput sequencing and use the magic algorithm developed in our lab to identify hits. And so if you look at the tumor growth, you can see here, compared to the nude mice, which is in this case where the tumor is growing very quickly, the tumor that are grown in the wild type immune competent mice is slower. When we have the vaccine ahead of time, it's growing even slower. And if we also add the PD-1 antibody, it's growing even slower. It's kind of pretty much controlled growth in here. And when we look at each of the previous, these three conditions, the CRISPR result compared to the nude mice situation, you can see in each case, the very top gene hit is COP1. Basically knocking out COP1, the tumor is growing slower in the immune-related mouse. And what is COP1? It turns out COP1 is a E3 ligase. And if you look at across all of TCGA tumors, it's like overall upregulated in many, many cancer types, suggesting that this is a cancer driver. And so we knock out COP1 in the cancer cell line, and we look at a lot of genes that are downregulated. They are related to macrophage chemoattractant, macrophage activation, TNF receptor, and cell adhesion molecules. This is on the RNA level. We also took a cytokine protein array to look at whether the cancer cells can secrete a different level of cytokines. And there are not so many upregulated cytokines. Instead, when we knock out the COP1, we see a lot of cytokines that are downregulated, which actually agree quite well with here. A lot of macrophage chemoattractant related cytokines are downregulated. And if we look at the COP1 gene expression in TCGA tumors, we have previously developed two different algorithms, timer for estimating immune cell infiltration levels. And you can see here that in many, many cancers, COP1 expression is positively correlated with the macrophage infiltration in the tumors in many cancer types as estimated by timer. We also have an algorithm TIDE which evaluates T cell dysfunction and exclusion in the tumors. And we have a computational model specifically for the M2 type of macrophage. So if macrophage is bad for tumor, the M2 macrophage is even worse. So initially you might have a monocyte or M0 macrophage, but it differentiates into M1 macrophage, which is more for recognizing or still trying to kill tumors. This is more for inflammation. But then the M2 macrophage is really for wound healing. It's actually pretty bad. If the tumor has more M2 macrophage, the tumor will grow faster and the T cells cannot really infiltrate the tumor. And you can see here, TIDE also predicted that COP1 expression is positively correlated with the M2 macrophage phenotype in many, many cancer types. And so we try to knock out the COP1 in the mouse, just this one gene. Previously it was a CRISPR screen, but now we knock out this one gene. And you can see here that compared to the control knockout, the COP1 knockout indeed in the tumor macrophage level infiltration in the tumor are much, much lower. To see what is the potential downstream target genes or the regulators that are related to COP1, because COP1 is a E3 ligase, it degrades some proteins. We want to see what are the transcription factors regulating these potential macrophage chemoattractant genes. We use a computational algorithm that we published earlier this year called LISA, which basically, if you have any differentially expressed gene set, we usually recommend having about 200 genes. And so if it's too many, you don't get general enough list. If too few, you don't characterize the differential genes very well. So we recommend roughly about 200 genes you input in here, and the LISA will use all of the data that we collected in this system database that we built to look at what epigenetic profiles, such as H3K27 acetylation or DNase, we collect a small group, or basically do feature selection to select the epigenetic profiles that represent this list of differentially regulated genes. Then it will use the transcription factor binding ChIP-seq data or the motif analysis to figure out what transcription factors are regulating those differential genes. And at the output, you will get two groups of genes, one group regulated the transcription factors related to the upregulated genes, the other related to downregulated genes. And so you can see here, when we run LISA on the upregulated genes versus downregulated genes, the downregulated genes, which are related to the chemoattractant, are enriched in some transcription factors in the CEBP family and also the AP1 family. We also evaluated the ADS family, but there's nothing significant. The reason for these three, we'll see later. So this is on the RNA level. We try to use the LISA algorithm on top of our system database to figure out what transcription factor might be regulating those differential genes when you knock out COP1. Next, we use a TAC-seq on the COP1 knockout cells to look at what peaks really changed if you knock out COP1. And based on the differential TAC-seq peaks, we can also use we have the system toolkit. It's another system function to see what transcription factors really have binding that overlap with this differential TAC-seq peak. And from there, you can see three families of transcription factors to be super enriched, just different members of the same families here. One is the AP1 family, one is the ETV family, and one is the CEBP family. So these three families are all important. And then third, we also use proteomics analysis to see if you knock out COP1, whether protein level, whether some factors are different. And this is the proteomics volcano plot, and you can see here, we see some CEBP, we see some ads, we see a little bit of AP1. So Jun and false are related to AP1, but they are not as significant. On the ADS family, we also have, besides ADS1, we also have ADS2 and ATV4. But remember, in the RNA data, we don't actually see ADS family to be related. So it seems like maybe the COP1 knockout influenced the ADS protein stability and ADS binding, but those ADS binding don't seem to have big influence on the downstream gene expression. And so basically at this point, we can see CEBP delta, it has good RNA evidence, it has good ataxic evidence, it also has good proteomics evidence to be an important substrate for COP1. Unfortunately, when we look at the known COP1 diagram motif, we don't see that diagram in the CEBP delta protein. So, basically, when you look at the C-terminal sequence of CEBP delta, there's not a known COP1 diagram on it. So how can COP1 degrade it? It's kind of quite puzzling. But interestingly, CEBP delta protein, even though it doesn't kind of directly recognize diagram on CEBP delta, it does recognize a TRIP2. So the TRIP2 has a very strong COP1 diagram motif. And previously, there's a paper suggesting that TRIP2 is a pseudokinase. It can be an adapter to link COP1 to CEBP for CEBP degradation. So we did the experimental validation. So basically here, COP1 recognized a COP1 diagram on the TRIP2. And then the triple can interact with CEBP, and then they will form a complex together, and the triple and CEBP can both be degraded. And so then Xiaoqing did the experimental validation to see whether these three really co-IP. So indeed, COP1, TRIP2, and CEBP delta, they can really co-IP. So we use each as a bait to fish out the other two. We can always see that these three form a complex. And if you look at COP1 knockout, we don't see that so see in this case the COP1 when it's knockout, it does not change the TRIP2 or CEBP delta expression. In fact, CEBP delta RNA level might even be a little bit lower. But interestingly, when you look at the protein level after a COP1 knockout, so in this case we use CRISPR to knock out the COP1 in two different cell lines. In each case, you see much stronger TRIP2 protein stability and also much stronger CEBP delta protein level. And so we also look at single cell data. This is using another database called TISH, where we have 76 different tumor single cell data of around 2 million single cells. And we ask in the macrophage, what are the genes that are correlated with the macrophage chemokines, or whether CEBP delta is related. You can see there's a very strong negative correlation. We also see a little bit of false, and we suspect that actually CEBP delta and FOS1 might have some interactions. And if you look at, for example, the CEBP delta binding in the cystrome in the public domain, you can see in this case CEBP delta also co-bind with STAT1 and RAL-A. This is part of NF-kappa B, and in this case, it regulates the expression of CCL2, which is a chemoattractant for macrophage. And you can see here, we can see after the ataxic, control, the COP1 knockout CEBP, the CEBP peak here is much stronger, and also the promoter of this gene become much more open, and that's how CCL2 can be upregulated. We have other cases for other chemoattractants for macrophage as well. So this is just one example. And so you can see then we try to knock out the COP1 individually and look at its effect on the tumor growth. Interestingly, when we look at the nude mice, where the mouse does not have an immune system or T cells, the tumor after COP1 knockout actually grows, even slightly higher, faster. But in the immune competent mice, the mouse have immune system, or the mice treated with anti-PD-1, you can see that when we knock out COP1, you can really see an effect of reduced tumor growth. We also use this COP1 knockout gene expression signature to look at the immunotherapy cohort that we have collected over the years. You can see in some of these cohorts, this COP1 signature doesn't seem to be significant, but anytime you see a significance, COP1 is always high in the non-responders. So when COP1 is low, potentially tumor have lower macrophage infiltration into the tumor, and then the patient will respond actually better to current immunotherapy treatment. And so in summary, basically, we use in vivo CRISPR screens to identify new immune modulators of tumor microenvironment. And we identify this COP1 whose knockout in the cancer cells can enhance anti-tumor immunity. Basically, COP1 can target CEBP delta via this 222 adapter and can degrade both of these proteins. And CEBP delta can actually activate the cytokine production and macrophage infiltration. And so when you, CEBP delta can actually suppress the macrophage infiltration, but if you have a higher COP1, it degrades CEBP delta, then macrophage can infiltrate the tumor a lot. And so we use this COP1 signature, and we found it to be associated with worse immunotherapy response. It suggests that COP1 is a very important target that you could use a drug to potentially inhibit in order to turn an immune cold tumor with a lot of macrophage into an immune hot tumor for T-cell killing. And so in this study, you can see Xiaoqing Wang is the experimental postdoc, but Colin and other computational member of the lab really help a lot in conducting data analysis and modeling. And so I want to thank members of my lab. You can see here, Colin did most of the computational work I presented. Xiaoqing Wang did most of the experimental work, and he's joint postdoc with Miles Brown's lab. We have many, many other lab members helping. And we also want to thank Richard teams who help us with the global protein stability assay validation from Steve Allergy's lab, and also Jin Liu from Wenyi Wei's lab for looking at protein fusion mutations. And also we're currently collaborating with Eric Fisher's lab to identify important targets for developing PROTACs. And I want to thank the funding agency. Colin is supported by the Damon Ryan Cancer Research Foundation, and Xiaoqing Wang is supported by Breast Cancer Research Foundation. So I think that's all for today. We also have many other computational resources. Some are tools that you can run on your own, including this tool, which is CRISPR screen analysis. And this is a bioconductor package, which is for the theme for today's meeting. And we also have a number of websites that you can just point and click and explore a lot of public data. So thank you very much.
Thank you, Shirley. There were a couple of questions in the polls. I think we only have time for maybe one or two. I guess the first one, BRCA1 was reported as a UPS with transcription factor ESR1. How does that impact drug targets and our understanding of breast cancer?
So far, ER is the easier case because among all transcription factors, the easiest to degrade are nuclear receptors. And so when you have BRCA mutation, ER is more stable, so it will drive kind of breast cancer growth. But in this case, you can use a hormone antagonist such as tamoxifen to inhibit ER. There's also other drugs that actually specifically degrade ER. Those are already FDA-approved drugs that are used. And so among all the transcription factors, hormone receptor, ER, AR, GR, those are the easiest to target. We didn't specifically, in this case, focus on nuclear receptor. We are a lot more interested in other transcription factors that normally are really hard to target with a drug.
Are 20K proteins generated from a single assay?
Yes, so basically you can do pooled oligosynthesis and you can synthesize all, 20,000 peptides in one order. You just send it to a Twist or Agilent, they will send back, it's like seems similar to whole exome sequencing capture assay. They will send you back a pool of oligos which contain the C-terminal end of all the proteins in the human genome. And then you just clone them into this GFP-RFP expression vector into lentivirus. And then you use the lentivirus to infect the cell, so you can do all the experiments in one Petri dish, and then followed by high-throughput sequencing. And so, Steve Allergy's lab previously published the C-terminus, that's one paper. The N-terminus is another paper. We built models for both. And recently, they are actually getting even the internal proteins. So they really try to look at every peptide sequence in the human genome, including the internal ones. So we might be able to find more diagrams, although based on our current modeling, most of the diagrams seems to be on the end.
In your talk, you show a lot of wet lab results. Did your lab do these, or can you give career advice to junior computational research, how to establish collaboration to validate computational findings? Yeah,
so for example, so in here, the GPS, so when we predicted novel diagram, Steve Allergy's lab were very interested in this assay. So when we told them we found novel diagrams, they were very happy to help us validate. And so I think if you have a common interest but complementary expertise, that's when they are more interested in collaborating. With the BCR-ABL, also is Wen Yiwei's lab, they are very happy. They're very interested in fusion protein as cancer drivers and the effect on diagram or protein stability. And so they did the experimental validation. With Xiaoqing Wang, you can see these two kind of is a pair. So Xiaoqing is a co-first author on Collins's paper, the computational paper, and Collin is a co-first author on Xiaoqing's COP1 experimental paper. So they really help each other. And so sometimes, we do some work to help our experimental collaborators, but then they also over time help us with our computational prediction, and those kind of trusts are built over time. And so then gradually, you help each other. And I think those makes a collaboration much, much stronger compared to a case where it's always the computational group providing help or sometimes even service to the experimental collaborator.
Thanks a million, Shirley. This has been a fantastic talk and so many different technologies. And I think it's really kind of the future where we do have to integrate a lot of multi-omics in order to advance biology and bioinformatics. Thank you, Shirley.
All right. Thank you. Enjoy the rest of the meeting.
Thank you.